Okay, so we'll be doing prions, our first lecture. We'll be covering the characteristics, transmission, pathogenesis, clinical manifestation, diagnosis, treatment, and prevention. Okay. So I don't know if you've heard the word prions. So prions are not our ordinary viruses. So this is a table showing the difference. Okay. So particle containing nucleic acid. When we say nucleic acid, we mean, is it either DNA or RNA? For prions, they don't have any of those. But for the conventional viruses, for example, HIV, we know it's RNA. We know hepatitis B, we know it's a DNA virus, like that, all those viruses. So prions does not contain that. Particle containing protein. So in prions, yes, but it is encoded by cellular genes. I'm going to explain what that means later as we continue. But for conventional viruses, we know it is encoded by the viral genes. So what happens here is the virus has specific enzymes that encodes its genes, inactivated rapidly by ultraviolet light or heat. So prions is very unique. So use of formaldehyde, use of all these techniques that you've learned, ultraviolet light, it does not inactivate it rapidly. So you have to do that for a very long time in order to inactivate it. But for the conventional viruses, it is easily inactivated. Appearance in light microscopy. So for prions, the appearance is filamentous or amyloid-like. We're going to see the picture. For conventional viruses, it depends. It can be icosahedral or helical in shape. Infection induces antibodies. So prions, very unique. The immune system is not activated. This is because the prion protein is very similar to the protein we have in our body. So no, but conventional viruses, yes. Infection induces inflammation. As I've said, the immune system is not activated, so there won't be an inflammation when you're affected by these prion proteins. But for conventional viruses, yes. So the prions we have human prions and animal prions. All of them cause something we call spongiform encephalopathy. Spongiform, so from the word spongiform, spongy, so it will cause spongy appearance on your brain, okay? Encephalopathy, meaning it is affecting the brain, okay? So we have CJD, Creutzfeldt-Jakob disease. This is the most common one. There are different types. There's familial CJD, variant CJD, as we have seen here. We also have fatal familial insomnia. We're going to talk about it. This one causes insomnia. We have Kuru, which is a variant of CJD. And then we have GSS, which is not that common. Gerstmann, Stroessler, Schickenka syndrome. Then for animal, Bovine Spongiform Encephalopathy, BSE, I'm sure you've heard of this. They call it the mad cow disease. And then we have Scrapie. So Scrapie is the version of mad cow disease, but in sheep. And then we have chronic wasling disease in mules or deers, okay? So all these prions cause spongiform encephalopathy. So classification, we can classify in terms of familial stroke hereditary, sporadic, meaning they come out of the blues, and then infectious. So prions, what is the definition? Why is it called a prion? So prion, it is small, proteinaceous, infectious particle. That's where we get the name prion, okay? Particles that are resistant to inactivation by producers that usually modify or denature proteins, okay? So this is the picture. So in our body, we have prion proteins, okay? These are the normal prion proteins. They're usually alpha helical sheets. You see here how they are surrounded like this.
Yeah.
Usually alpha helical.
Hello.
Yes?
Could you go back to the previous slide that
This one?
about there's something that you talked about the
This one or
The
this one?
previous one?
This
Yes,
one?
this one. Yes, that one. Could you?
Okay. Do you have a question?
Yes, there's something that you talked about the prions, the reason as well they are being called. So it kind of passed me, Kiasi.
Oh, okay. So they're small proteinaceous infectious particles. So from this word, proteinaceous infectious particles that are usually resistant to inactivation by procedures that usually modify or denature proteins. So I wanted to explain why they're called prions. So I was showing you this picture. So we have normal and abnormal prion proteins. So in our body, we have normal proteins, normal prion proteins. Now, if you look at this, it's called alpha helical sheets. They are alpha helical sheets. This is the pattern, as you can see, the one in green. But what happens is the abnormal prion proteins, they usually are misfolding, okay? You'll do this in biochemistry. There is a misfolding of these proteins. So as you can see, this blue part, it misfolds. So when it misfolds, it converts this to a beta pleated form, okay? Alternative folding pattern, or PRPSC. So PRPSC means the beta pleated form of this prion protein. And what is unique about this one when it misfolds is it cannot be broken down by proteases. You know, in our body, we have certain enzymes that break down abnormal proteins, okay, during the cell division process. But now here, it will not be broken down by those proteases. Okay, now this is what we call the beta pleated form. So now this is what we call the prion, the abnormal prion. Okay, have you understood? So characteristics
Yes.
of prion, they are made of protein, as you have seen there. The protein is the alpha helical sheets. Resistance to inactivation. So as you have said, after the misfolding has occurred, it becomes resistant to inactivation by both protease enzymes and the ultraviolet light or heat. Reproduced by converting normal protein to abnormal protein. Now, this is the process that occurs. Once you, let's say, ingest a protein, now goes to the blood-brain barrier, reaches the brain, it now converts normal protein to abnormal protein. Do not induce an immune system because they cannot be inactivated. So there is no inflammation, no immune system activation. Cause vacuolation of neurons leading to the spongiform appearance. So we are going to see, let me explain this point. So as I was saying, this is the alpha helix, the normal prion structure that is found in our body, but now it is converted to the beta chain pleated form. So there's a misfolding here. So one amino acid like this one will be misfolded and another amino acid will occur here, as you can see here. Okay. And then protein structure, these are the normal protein structure. For example, in insulin, we have a beta sheet, alpha sheet, beta sheet. Ribonuclease, we have an alpha helix. So you can imagine if misfolding occurs, is what will get the prion protein. So before I explain this, I wanted to show you guys this spongiform appearance. So what happens is, when the prion protein, let's say you ingest the prion protein, it finds its way to the blood-brain barrier, crosses over the blood-brain barrier, then it enters the brain. When it enters the brain, it will now trigger apoptosis. You know, apoptosis is cell death. Basically, the cell is going to burst and die in the brain. When that happens, cysts will be formed. Can you see these cysts? So this cyst is what we call the spongy appearance. Can you see here? This is the normal brain matter. As you can see, there are no cysts, but you can see here. Now that is what we call the spongiform appearance. Okay. So these are the cellular proteins, the ones that are normal, and these creepy ones. So which is the infectious prion. So cellular, as we had said, predominantly alpha helical in terms of structure. These ones are predominantly beta sheets. They're called beta pleated form. These ones are soluble. These ones are insoluble. These ones are non-infectious. These ones are infections. And then these ones are susceptible, resistance to proteinase K. So these are the proteinase enzymes. This one is resistant. Okay. Have you understood? So we're getting to see the abnormal ones. These are the characteristics of the abnormal prions. Transmission, as we had said, we have sporadic, iatrogenic. So iatrogenic means it is associated with the hospital. So for example, during blood transfusion, when you have these prion viruses in your blood, during blood transfusion, I can get them. During organ transplant, okay, most common is, for example, corneal transplants. Corneal transplants are, the cornea is the outer covering of the eye. So when you have certain conditions that make your cornea weak or very thin so that you cannot see, we usually do corneal transplants. So when you do these corneal transplants, you might get this prion infection. Hereditary, there's some evidence that it is passed from mother to child. Then ingestion. So ingestion, this is the kuru. You remember we had mentioned a strain of the prion. It's called kuru. So when we, especially when you practice cannibalism, cannibalism is where human beings eat human beings. So when you eat the meat of human beings, so how it was discovered was, there was, in a certain country, it's called New Guinea, they were eating people. And when they started eating people, they noticed that the prion infection was quite common there. So they concluded that it can be spread through cannibalism. And then beef, if you eat the meat of a cow that has mad cow disease, remember that we have said the bovine spongiform encephalopathy, you will get the variant CJD or the variant Creutzfeldt-Jakob disease. Then if you eat a sheep that has scrapie, so that is mutton, you'll get the prion infection, okay? Remember here, we had mentioned them here. Yeah, so if you eat an animal that has this bovine spongiform encephalopathy, you're going to get the prion. If you eat an animal that has scrapie, or if you eat a deer that has these animal prions, you're going to get it. Then the kuru is the one that we said it is spread by cannibalism. Okay. Pathogenesis. So this thing has a lot of biochemical names that you may get confused. But the most important thing I want you to remember is the prion, the normal prion we have in our body is going to be misfolded. So you're going to ingest a prion from, let's say, from an animal, from a human being, or you're going to be spread through blood transfusion. Then this prion protein will make its way up to the blood-brain barrier, crosses the brain. When it reaches the brain, what happens is it's going to cause misfolding of the prion proteins there. And when that happens, it's going to trigger apoptosis, leading to cell death. And when cell death occurs, there's going to be formation of cysts, as we had mentioned here, cysts in the brain. And then this is what we call the spongiform appearance on the brain. Now, this is what is going to cause spongiform or vacuolation encephalopathy. Okay? So to read this, normal cellular proteins is found on the surface of the brain cell and other cells. Infection or mutation of PRNP gene on C chromosome 20 can cause PRCP to misfold into PRPSC. So this is the beta. Basically, what this line is saying is if you have a mutation in the PRNP gene on chromosome 20, it will cause misfolding of the prion protein to abnormal prion protein. Basically, the alpha helix will change to the beta pleated form. So the beta pleated form dissociates from the cell membrane. The beta sheet causes more protein, the alpha helix, to misfold and dissociate from the cell surface. Accumulation of the beta forms proteinaceous plaques between the brain cells. Then aggregated beta sheets is then internalized into cell, leading to cell death, leading to spongiform appearance. So this is the theory, but I had explained it. So have we understood how spongiform occurs? Have you understood? Or you want me to repeat?
Yeah, can you repeat?
So I want us to start from here. One thing you have to remember, this is the normal prion. The normal prion found in our bodies. This is the abnormal one. So let's say, for example, me, I ingest meat from a cow that has prions. What will happen is the prion will find its way to the blood-brain barrier. When it crosses over, it will cause this normal prion that is found in my brain cell surface to be misfolded. Okay, biochemical reactions will occur, then it will cause misfolding. It will change from its normal alpha helix appearance to its beta chain appearance. And when this happens, this beta chain appearance of the prion will cause apoptosis or cell death in the brain. And when this cell death in the brain occurs, it will cause this cyst-like appearance or vacuole appearance on the brain. Now, this vacuole appearance is what we call the spongiform. It looks like a sponge. If you look at it, have you seen a sponge that we use to wash the utensils? It looks like this. So that is what will cause the spongiform appearance on the brain. Okay, is it clearer now?
Yes, thank you.
So these are the variants of the disease. We said Kuru, it's transmissible, stroke infectious, caused by ingesting or handling brain tissue, occurred in New Guinea tribes. So these are the ones that are practicing cannibalism. Then CJD or Creutzfeldt-Jakob disease, transmissible. We said iatrogenic through corneal transplants, brain electrodes, and growth hormone. Hereditary, that is a mutation in the germ cells. Sporadic means it just comes out of the blues. And then we have possibly a new mutation in the somatic cells, and this is the most common form. Then we have variant CJD, acquired by eating meat or nervous tissue from animals with mad cow disease. And then we have GSS, hereditary, mutation in germ cells, fetal familial insomnia, mutation in germ cells. Then we want to talk about the clinical features. So what happens after you've had conversion of your brain tissue to the spongiform form? Number one, it usually has a long incubation period. So for example, it can take 10 years because your cells are continuously dying, slowly and slowly. And it is very slow and progressive. Ultimately, it will be fatal since you will have, by the time the 10 years or 15 years have elapsed, you'll start showing it will be fatal because more brain tissue is dying. Cognitive impairment, what this means is you'll have signs of dementia. So dementia are things like, if I ask you, you will lose sense of time. So you'll be asked, what time is it? You'll say, I can't remember. You lose sense of place. Where are you? Maybe you're at home, but you cannot remember. And then ultimately you lose the sense of who you are. It's called sense of person. So if I ask you, Steve, who are you? You don't know who Steve is. Or if I call you, you'll be like, oh, I'm Steve. Okay? So that is what you call the cognitive impairment. Very progressive. You lose sense of time, place, then finally person. Okay? Ataxia. So this is a movement disorder. How I describe ataxia is, have you ever seen a drunk person? How they stagger. Okay? So that is how ataxia looks like. It's a movement disorder, how you cannot walk properly. So ataxia, as you can see, it usually affects the cerebellum, whose cerebellum is responsible for movement. And then we have myoclonic jerks. So we call them myoclonus. Myoclonus are these repeated movements. Okay. So if it's your hand is going to move repeatedly at a very fast rate, rapid repeated movements. And then mutism, you'll be unable to talk. Okay. And then the FFI, fetal familial insomnia. It was one of the, remember here we had mentioned it, fetal familial insomnia. So signs of fetal familial insomnia is insomnia. You will have insomnia. You cannot sleep. First of all, insomnia is defined as difficulty in initiating sleep. So you may be trying to sleep, but it's hard to initiate it. Difficulty in maintaining the sleep. So you might have no difficulty in initiating it. Let's say you sleep at 10 p.m., but now maintaining that sleep will be difficult. So maybe you'll be waking up 2 to 3 times a night. And then finally, difficulty in waking up. So you'll have a hard time waking up, or basically you wake up feeling very exhausted. So that is what is called insomnia. Okay. So this insomnia, it can get even worse, where the person will have difficulty initiating sleep for the whole night. They won't be able to sleep. And when you're not able to sleep for a whole night, what happens is you start getting hallucinations. So for example, you'll start hearing voices, you'll start seeing things that are not there, okay? Those are the types of hallucination. Dysautonomia, so your autonomic nervous system will be affected. And then motor paralysis, basically you'll be paralyzed, you'll not be able to move, okay? I'm seeing a question. Does it affect all the brain parts or a specific lobe? So it usually starts with specific. As you can see here, we are having cerebellum manifestations, the ataxia, myoclonus, and then cognitive impairment. So this is at the temporal lobe. So it goes progressively until it affects the whole brain, okay? Then for diagnosis, there's no specific diagnosis that we have. The definitive diagnosis for prions is usually a brain biopsy. And you know, in a brain, you cannot do a brain biopsy. You have to wait for a postmortem or postmortem or autopsy. We call it autopsy, where we're going to see the spongiform appearance on the brain tissue. Yeah, as I had shown you. So this is the brain appearance on a dead person. So it's very unfortunate. So diagnosis, we have to exclude differentials, diseases that present in a similar manner. Then we can do CSF analysis. We do a lumbar puncture. We'll find 1433 protein or tau proteins on the cerebrospinal fluid. EEG, MRI, I mean, they have low clinical use. So the only definitive diagnosis is the brain biopsy. Okay.
Hi, sorry.
Sorry, yes?
What is the EEG?
Okay. Oh, sorry. So EEG is, I don't know the full name, but basically, what usually happens, let me check I don't want to butcher it. EEG.
It is electroencephalogram.
Yes, and electroencephalogram. What happens is, if I have a picture, you put nodes, the electoral nodes. I'm sure you've done physiology. Do you remember the ECG? When you did CVS physiology, ECG, when you are checking the electrical impulse on the heart?
Yes.
Yes. So it is the same principle. Same principle we have here. We do the same thing. We put the electrode nodes on the head. We are monitoring the waves. You know, when you're sleeping, there are brain waves that are, don't say excreted, but are given off. Okay. Now these brain waves are the ones that we are going to analyze and see. But it has very limited clinical diagnosis. Okay. But that's the principle. The same principle we used for ECG is the same principle we're going to use here. But now this is electroencephalogram. Okay. I'm seeing another question. What is meant by exclude differentials? So when we say exclude differentials, it means we want to exclude similar diseases that present like CJD. So what that means is a patient will come with these diseases, these clinical features. Most common is dementia. So when you see a patient who has dementia, you don't start thinking CJD as your first differential or as your first cause of that dementia. There are very many causes of dementia. The most common is Alzheimer's. I don't know if you've heard of it. Alzheimer's disease, it occurs on old people. where they become senile and then they get dementia. So that is something we should exclude. So Alzheimer's has a certain way we exclude it. Lab test for Alzheimer's. And then we have some other causes of dementia. We have Lewy body dementia, Huntington's dementia. These clinical features can also present in infectious causes. For example, if you have like a viral encephalitis caused by herpes simplex, okay, or any other autoimmune disease. So that is what we mean by excluding differentials. We exclude each and everything so that we remain with one disease, okay? And then treatment, as we said, there's no treatment. We usually just give supportive treatment, palliative care. Palliative care means we put the patient in a good state before they die, because eventually they're going to die, but we don't really prolong their life, but we make their life to have good quality. It's good quality care. Then prior disease are invariably fatal within months to years. Okay. Prevention. Remember we said it is not prevented by normal sterilization. So the sterilization of surgical equipment should be for 1 hour, 134 degrees, autoclaving and sodium hypochlorite for 1 hour. You can see, because they are not easily damaged by sterilization. Then screening of tests, screening of blood and organs. So before blood donation or transplants, then we ban on meat and bone meal in animal feed. Do not feed cows to cows. So this cannibalism. And then if any animal shows features of mad cow disease, we have to kill it and burn it or bury it, okay? Do not eat meat from such animals. Okay, so this is a question for you guys. One of the following is true about abnormal prion proteins. They strongly induce, okay, answers on the chart. Good. I'm seeing you understood. So choice A, strongly induce the host immune system. We said it does not. So basically, you'd ask yourself why. This is because these prions, the immune system, you know, the immune system, for the immune system to be induced, it has to think something is foreign. But now these proteins, these prion proteins, as we said, they are not foreign, so the body will not induce an immune system response. Reproduce through budding. They don't reproduce through budding. We didn't mention budding anywhere. We said they propagate by inducing normal proteins to misfold into abnormal prions. Contain RNA and DNA. This was one of the first things we mentioned when we started. They do not contain any RNA or DNA, any nucleic material. And then cannot be inactivated by formaldehyde, as we said. So we have to use autoclave and sodium hypochlorite for 1 hour. Good. This one. Regarding prion-mediated diseases, which of the following is most accurate? Okay. Answers in the chat. Mm-hmm. Some people are saying A. Any other answer? Navia? Janet?
Is A the right answer?
Do you think it's A?
Yeah.
Okay. Janet? Janet hasn't spoken since we started. Is she there? Janet, are you there? No, I think she's not there. Okay, no worries. She might have left. Oh, she's here. What do you think the answer is? So let's start from down while we eliminate, okay? In CJD, only neurons latently infected by JC virus produce the prion filament that disrupt neuronal function. So is there anywhere we have mentioned the JC virus? There is nowhere we mentioned the JC virus. Yeah? Is there anywhere we've mentioned JC virus?
No.
There's no way I've mentioned JC virus. The JC virus, I don't know if you have done it. It's a DNA virus. I don't know if you guys have done it.
No, we have not done it.
Or you have not done it. Okay. It's a very different type of virus. Okay. It usually causes something we call PML, progressive multifocal leukoencephalopathy. Let me write it for you guys down. Called PML. Okay, that is the JC virus. There's no way we have mentioned JC virus. It's under polymoviruses. Okay. So you'll see this virus is quite common in especially HIV patients who are immunocompromised. They usually get PML. Okay. So there's no way I've mentioned CJD and JC virus. They are very unrelated. Okay, let's go to the next choice. C. Kuru is a prion-mediated disease for which the diagnosis can be confirmed in the lab by a fourfold or greater rise in antibody titer. Have we mentioned anything about that? We said, first of all, prions do not induce an immune response. So there's nothing about the antibodies here. So C is wrong. Then variant CJD is a disease of cattle caused by ingestion of sheep brain mixed into cattle feed. So do we ingest sheep brain mixed into cattle feed? No. What do we ingest? Affected cow, isn't it? And then prion-mediated diseases are characterized by vacuoles in the brain called spongiform changes. Correct. So that is the answer. Any questions on prion?